You starting to think about this?
Good afternoon, everybody. Yes, today's topic is going to be about how to get AI ML software as a medical device FDA cleared. My name is Yujan. Thanks for being here today. First of all, also thanks to my wife and family. This time when I get back, I'll also change some dirty diapers too. So, my name is Yujan Shrestha. I represent my team named Analytics. We are 18 strong software engineers and also regulatory consultants. Here are some of our services. Our company offers software engineering services and also regulatory services. If you want to know more, our booth is right over there. You can see it from the auditorium. And also after this presentation, we'll be happy to take any questions at our booth. Thank you. Here are also some of our respective clients that some of them you probably recognize at RSNA. We've had the privilege of helping them. Just wanted to give it a shout out. So unfortunately, I only have 20 minutes for this conversation, and I can't really possibly tell you everything that we know in 20 minutes. What we're about to share with you is an average of lots of projects we've been through. It will probably get you 80% of the way there. 80% of these topics will apply to your device, but there's always that last mile problem with the last 20% that you should get some advice for. These are also highly opinionated. In no way is this an endorsement from FDA. So I want you to take away from this conversation rules of thumb, nuggets of wisdom, 80-20 trade-offs, guiding principles, and the way that we have found success at Analytics doing this process. So your mileage might vary. Please don't take this and plug it into ChatGPT. We've seen that happen. We have to clean that up. So that's not going to work, unfortunately. So the golden road of medical device development might sound cliche, but I think it's important. You should design for others as if you would design for yourself. Then after that, find the least burdensome pathway to market. First question that we get a lot: is being statistically powered enough to prove generalizability, enough to get FDA cleared? And the answer is no. Statistical power on its own does not prove that your device will also generalize, because it does not account for biases within the data, things like age, race, different types of devices. And what FDA is looking for is reassurance that your device, trained on your training data, tested on your test data, will generalize when it gets into post-market. So unfortunately, sample size, although that is one facet of what you need to prove, it's not the whole story. So then how much data is enough then? This is a very age-old question. And our rule of thumb on this, for non-CADX, so non-computer-aided diagnostic devices, 200 subjects seems to be the sweet spot. If you need to do manual annotations, you need 3 readers to make sure they're US board certified readers. Alternatively, you can have alternative ground truth sources like biopsy results, head-to-head predicate results. That can also be an option for you. My recommendation for how you should allocate your budget for testing data: use 80% of your budget for the first big data pool. That should capture a lot of variability in your target data, but it's not going to capture all of it. That's what the remaining 20%, I like to fill in any gaps in that. So if there's any demographic gaps, if there's any gaps that perhaps you're missing a type of scanner manufacturer or magnet strength or whatever, that last 20% of your budget could be used to fill in those gaps. And the idea here is to identify potential sources of variability that could impact the actual performance of your device. Things like imaging protocols, acquisition systems, BMI, if you're doing like whole body quantification, for example. That's the idea. Make sure 50% of your data is from the US. And also, if you need to do a multi-reader study, make sure you're using at least 12 readers. That's been our kind of best practices. For your training data set, these principles are less required. Your training data set could not necessarily have all these, but for your test data set, that needs to be locked down pretty well. Next question. So to file or not to file, that is an excellent question. How to handle ongoing changes. So there's draft guidance on this from FDA. or not draft, it's guidance back to you, but it hinges on your interpretation of the word significant. Is it significant enough? Is it significant enough of a change to just retrain your algorithm? In our interpretation, that does not constitute a significant enough change to require a new 510K application. If you want to play it safe, there are other avenues like a special 510K and also a PCCP, which is a predetermined change control program. But if you're adding something new to your device, like a new disease condition or a new type of imaging modality, those probably are considered significant changes by the word and would require a new traditional 510K. Unless you've already done a predetermined change control program with FDA where you pre-brokered a deal. It's kind of like a pre-cert program for changes that you know that you're going to do. So you can do that in your pre-market submission. We have an excellent article on our website. If you would like that, please go to our website and review, or again, go to our booth for the post-show questions and answers. So performance targets is also a huge source of concern. Our kind of rule of thumb again, if you're doing diagnosis or detection, shoot for an AUC of greater than 0.9. If you're doing a segmentation target, shoot for a DISCOR greater than 0.8. For any sort of continuous values, shoot for plus or minus 5% of that value, 95% limits of agreement, confidence interval for that. However, you still need to justify to FDA how you got these values in the first place. And in our opinion, there's 3 tiers of evidence that you can use to justify these values. The first and most preferred tier is you can get your predicate device and see what they did. If they publish performance targets, that's usually a very strong source of evidence to justify why you chose the targets you did. Failing that, you can also check on like the academic literature to see if there's any papers published on like similar algorithms that have been done, and you can use that as further evidence to justify that your targets are meaningful clinically. And finally, if you have a completely new quantitative imaging value that doesn't have a precedent, you could just measure your device on like a pilot set, like a tuning set, and then you can set your targets to kind of give yourself a little bit of an error margin. But that's the least of the preferred. Also outline the impact of false positives and negatives. So if there's a false positive, if you claim that there is a lesion and there isn't, what's the output of that? And that could be different based on what the clinical output is. What FDA is really looking for is the risk that is associated with your device. So at Analytics, we do R&D and also D&D to on these machine learning algorithms. So our process, we don't transfer the algorithm out of the R&D step until we know with some degree of confidence on the tuning set that we're going to meet those aforementioned targets. And we will continue iterating on R&D until we do meet those targets. Now, unfortunately, not all devices have an unlimited budget or unlimited time frame. So if you, at some point you'll have to get to a point where there's no more budget for another R&D round, and in this case you'll have to make some trade-offs. Trade-offs might not be as bad as you think. For example, a bounding box to outline the overall location of a tumor could be just as good from a sales and marketing and also clinical need standpoint, but it could be 10 times easier from like an engineering standpoint. So you can make these little micro trade-offs as you go, and to find that sweet spot into creating something that the market wants at a reasonable price point. And one other common thing that happens, there could be a subgroup where your algorithm performs very poorly, but that subgroup is something that's not really clinically meaningful. That subgroup is something that's not meaningful from like a business perspective either. So you can add in your labeling that your device doesn't support it, or it supports it with less accuracy, and give ground there where you can then save ground on some other topics that's needed. So it's important, kind of going back to that same point, it's important to have a cross-functional team. And at Analytics, our process is to have, at the end of every R&D sprint, we have almost like a tumor review board. We go over qualitatively how is the device performing, and we put that in front of clinicians. We put that in front of marketing and regulatory stakeholders, because these folks know what the market wants, what will sell, what's going to be easy to make, and regulatory will know what's going to blow up the study design. So those viewpoints put together will give you a much better loss function than just a DICE or an AUC alone. Because there's a lot of nuances that can't be captured in the DICE score. Like for example, if it's important to you to avoid blood vessels, that like a DICE score of 0.9 might be actually perform worse than a DICE score of 0.8 in that case. You won't know unless you look at it. And what we've seen, we've also designed our process to avoid this, is sometimes engineering will be in its own silo. They'll be chasing a DICE score, but in reality you'll end up making a product that no one wants to buy, and you'll end up taking 10 times as long and costing 10 times as much. Also, biostatistics, we prefer to have biostatistics with some amount of programming experience so that those can be fully automated, because you'll be doing a lot of subgroup analysis as part of this process. When you get your feedback from FDA, almost always there's a subgroup that you haven't done that you'll need to do. So having some quick mechanism to repeat analysis in a subgroup, I think that will save you a lot of headache. So do I need a pre-sub? Yes. When's a good time for a pre-sub? We like to do it right after the algorithm prototype is done, so we have a good idea about the overall performance of the algorithm, and we know how to design the study in order to go into that pre-sub. And a consultant that has some software development experience is our preference, because that consultant should be able to answer a lot of questions that you don't have to go to FDA for. Things like cybersecurity, software validation. These are questions that don't need to go to FDA for pre-sub. So you should ask the high-risk stuff only, which usually for us is going to be the study design. The study design, to execute the study, could be a costly mistake to get wrong. These studies cost order of 150 to $1 million plus, so that's a high priority in order to ask FDA. It's important to communicate your algorithm design to FDA. FDA is very technical. They want to know how the algorithm works, they want to know all the steps involved, and they want to know the failure modes. So let's make their life easier by breaking out into different categories. And here's a block diagram key that we've developed. You can also look at more of this on our website. We like to split out the non-machine learning components from the machine learning components. This helps to fully communicate to FDA where are the high-risk areas, and they'll focus on the machine learning components whenever they look at this. They need to know that what other components are there to make sure there's nothing hiding in there in the weeds. So if you give FDA a lot of uncertainty about how your algorithm works, you're going to get a lot of uncertainty back. Or worst case scenario, which we've seen a lot, is if there's a lot of uncertainty in FDA, they're going to give you the most conservative answer. And that conservative answer usually ends up costing you a lot more. So again, here I definitely like to map out the inputs and the outputs in this kind of diagram here. And this allows FDA to see quickly what's machine learning, what's more kind of traditional image processing. And we have some more research on our website if you want to see that. So if you need help for a pre-sub, please stop by our booth. We'd be really happy to help you out. You only get 1 hour with FDA, so every second counts. And again, prioritize that study design. I really like this diagram from Dr. Keith Trader back at the FDA Advisory Committee meeting a couple of weeks ago. It clearly shows the level of risk involved in different types of AI devices, the different types of CADs over there. And you can see a clear demarcation, what's in red and what's in green. The red requires a multi-reader study, which that's the ones that cost lots more money than this standalone performance test. So I'd like to break down what risk means to FDA in this case. I like risk as kind of like the how much confirmation bias that you are injecting into the practice of medicine. And there's two vectors to this. There's location bias, you're pointing a clinician to a specific spot, and there's diagnostic or clinical bias, where you're telling them or suggesting to them what to do next or what the diagnosis is. Those are two different axes, and you have a magnitude between those. Quantitative imaging devices like product code LZ, QIH has the lowest magnitude, where they only automate a process, they don't introduce that much confirmation bias into the practice of medicine. A computer-aided detection device, on the other hand, introduces a large amount of confirmation bias, but it's usually in the location axis, where you're pointing the clinician to a specific spot, where you're focusing their attention there. So they might miss other spots. Or if you tell them nothing's there, they might not really go into more detail. So that's the risk that FDA is looking for, is the confirmation bias that you have injected. Is that beneficial to practice medicine or not? So on the other hand, computer-aided diagnosis, that's also injecting a large amount of confirmation bias, but it's going in the other direction. It's going in the diagnosis or in the managing clinical condition direction. And these two directions are not parallel. So you can have a device that does both. Or for example, if you have a device that will identify lung nodules, that's a CAT-E. If you have a device that does lung nodules and tells you if it's also cancer or not, that's a CAT-E and a CAT-X. And finally, there's computer-aided triage. So this is the most restrictive of these here, but it could offer a easier pathway to market because the study design is a little bit simpler. But you can't inject confirmation bias in any of those two axes. You can't inject it on where the finding is. You can't inject it on what to do next or what the diagnosis is. You can only prioritize what images to review. So that may or not work for your business use case. So for CAT-E design, here's an example of how a study is going to be done. First, you collect your data using the strategy we talked about. We like to use two teams of two experts to do the annotations. There's going to be some sort of technician that does pre-reads, and then we have a US board certified clinician that then does the QA step and does any editing. Do your ground truth, and also find any areas where the two teams of two disagree, and also adjudicate those ground truths. And importantly, only run the standalone performance study when R&D is completed. So when the engineering team feels satisfied with the current state of the progress and they're ready to lock down the weights, they've run it on the tuning data and they're passing the metrics there, then run the standalone performance study, and only then after that run the multi-reader study once you've passed the standalone performance test. It could be an expensive mistake to run the multi-reader study too early. So multi-reader study could fail for one of several reasons. The first reason, your algorithm just might not perform that well. And if you did your standalone performance testing first, you would be able to find that out before doing this multi-reader study. So in this case, standalone performance testing works well, or doesn't work well, and your multi-reader study won't work well either. Also, there could be subtle tweaks to your UI that could really improve this multi-reader study. Perhaps the UI, your tumors are just too transparent, it's really difficult to see, or small lesions are really difficult to see, so you might want to put an arrow there instead of just a little dot. Things like that, you would see a high standalone performance test than a low multi-reader study output. Also, some readers could just be skeptical of AI and may not really want more confirmation bias or want additional information. In that case too, your standalone performance test would be high, multi-reader study would be low. And there's typically a 30-day washout, the standalone performance test, which you can see why this one is much more expensive than the previous slide that we did. So some cybersecurity, you can learn more about that on our website there. Basically, the gist of it is be sure to involve your software engineering team in this. Our engineers are typically, if we can't come up with a low-hanging fruit to properly solve cybersecurity issues, there's something deeper, or going a little bit too deep into that topic, and you should be able to justify to FDA when you don't need to do that. Human factors. If you don't have any critical use cases, you don't need it. User needs validation, survey, simple, if it's a low-risk device. Automated tests, you should use those. Sorry, I'm running out of time, so I'm gonna go through some of these here. So some things about Gen AI and foundation models. Currently, nothing Gen AI has been cleared by FDA. And just because your device does the same thing that your predicate that doesn't use Gen AI, it doesn't mean it's going to be substantially equivalent. Jury's still out on that. Here's some tips and trips for Gen AI stuff. Clear indication, don't do everything under the sun. Constrain your inputs and outputs. Always keep it so that so the user can't just use you as a chat GPT, where you have not cleared indications that are being used. Include things like anti-prompt injection and anti-jailbreaking attacks. And also just a little plug, also pick a consultant with strong engineering experience like us. So yeah, thanks for your time. Hope you learned something. And again, if you did, please continue the conversation right over there at our booth. Thanks a lot.